Alright, dear students, I'm Shabana here with a series of lectures on biology. This topic would be on inheritance, which is one of the chapters in your O-level Cambridge biology syllabus, right? Now, in inheritance, we will also have to look at genetic crosses. Now, what do you mean by genetic crosses? Well, genetic crosses will give you the probability. Let's have a look at this. So we will look at a few definitions and then we look at how do we sort out the genetic crosses. Probability of a certain trait, all right, probability of a certain trait being transferred from one generation to the next, from one generation to the next. Right, now comes the question about what is a trait. So let's have a right look at this. Traits is another word. It's just the same thing. It's just another word for the characteristics of an organism, right? Characteristics of whatever organism you're trying to study. So we're looking at the different characteristics. Now, for example, this could be hair color, hair texture, the blood group, all right? And for plants, it could be the leaf size, the plant size, the height of the plant, you mean. So, for example, this could be hair color, or this could be hair texture, right? And so many, for example, also for plants, we can talk about leaf size and maybe the number of stomata and so on and so forth, right? Now, if we look at these traits, then we'll have to study about genetic crosses. How are they transferred? So the first one that we look at is when we take only one trait at a time. Now, the technical term or the scientific term or the biological term for that is monohybrid crosses. Now, monohybrid, what is the literal meaning of the word? The monohybrid cross means, mono means one and hybrid means a cross or a combination. So we can say the transfer of one trait, all right, and what will probability will be in that organism. Transfer of one trait, you can add the word only here, right? Now, the different types of monohybrid crosses in your syllabus are, I'll make a list and then we'll go through them one by one. Now, the first one is what is termed as complete dominance. Now, those of you who want to have an interest in, you might say, the history of genetics, then Mendel was a mathematician and he introduced the concepts of genetics and all that, the genes and all, and he gave in these, you might say, genetic crosses, okay? So the first one is complete dominance and another one would be incomplete dominance. All right, so the first one is complete, the second one is incomplete dominance, and the third one is co-dominance. So we will be looking into each of them, but the first one is this one, so you need to understand this. Now, after that, we can also look at test crosses and then determination of gender, or we can also look at sex determination, and something linked with this would be sex linked inheritance, right? Okay, so the first one we're going to look at is complete dominance. Okay, so the first type of crosses is complete dominance, and as the word suggests, complete dominance, and as the word suggests, there will be two alleles of a gene and one will completely dominate the other, right? So what do you mean by that? Just have a look at, let's see. All right, so we go back to Mendel, who is said to be the father of genetics, and Mendel studied the pea plant. All right, so we'll take his example. It's easy to understand also. We'll take the pea plant and, for example, he took at least 6 traits of the pea plant were studied and he made some laws and some genetic crosses, right? So we'll just take one of those traits and that will be the height. So the trait was height of the pea plant. Now, why was this easy to study? Because the pea plants had only two variants of the height. Either they were tall or they were short. So there were two variants. Either the pea plant was very tall or it was very short. The short one is said to be the dwarf variety as well, right? So the taller ones would mean that the pea plant was actually approximately like said to be around 6 feet, whereas the dwarf variety was very, very short and it was around 2 feet only, right? So you got this. So let's have a look. Now, to understand or to draw genetic crosses, we need to learn what is known as the language or the symbols of genetic crosses. And what do you mean by that? Let's see. All right, now first of all, so when you look at height, now for example, Mendel grew 1,000 plants, which is a supposed approximate number. He grew 1,000 plants, and out of them, what he discovered was most of them were tall, and the rest of them, or the less common trait, was the dwarf variety or the short variety. So tall was more common and short was less common. So what he did was he gave them a name. The tall one, the one that was more common, that variety or that gene, he said will be considered as to be the dominant trait. All right, and the less common one is termed as the recessive trait. All right. Now, when we look at these genetic crosses, when we draw them, we're going to use some symbols. So what he did was, number one, for the tall one, which was the dominant variety, we will use the symbol, we'll take the first letter of the dominant trait and we'll write down that letter. This capital T represents this trait, as well as being capital, it shows that it is the dominant one. Now, what were the letters used for the short one, for the short variety? Now, what short form or what symbol would I use? Do I use S or do I do something else? Now, if I use S, it becomes too confusing. So, what I will do is I will only take the lowercase of the same letter, and that will be T. So, small t means that number one, this is a recessive trait. And obviously, it's recessive to the dominant one, capital V, T, meaning that this is the dominant trait, right? So, all genes, when we say that they are dominant and recessive, or there are two types, we actually mean, so chromosomes present in sexually reproducing organisms, the chromosomes that are present, they will be present in pairs, and these pairs are termed as homologous pairs, and I'll do a separate lecture on the concept of pairs, chromosomes, homologous pairs, and the genes and the alleles. So now what does it mean? So you have two chromosomes, and each of these, the homologous pair, each of them will have a gene at a certain place, which will be of the same type, of the same position, and that is why they are termed as homologous pairs. So whenever we look at the two terms, we mean these two variants. So this is like two genes, and there are two variants of a single gene, and these are termed as alleles, right? So one gene, there will be one gene, and one gene will have two alleles, right? Now let's see how will we write them down, the language that will be used, and we can understand this. Now, so we're going back to symbols of genetic crosses and how do we write them down. So now the first word is going to be, there's going to be a lot of terms in this chapter, and as you go along, obviously there's going to be a lot of repetition. So as we go along, we'll keep on repeating the words again and again, and you'll have a good idea. Now, What are the possible, you might say, genotypes? So when we write down the genotypes, we put down both the alleles together. Now the genotype could be that there is going to be capital T and capital T together, and to understand that, you can make a diagram and see that in the homologous chromosomes, the genes that are going to come, for example, from this side, from this side, and as you know, these two homologous pairs, one is coming from the male gamete, and the other one is coming from the female gamete. So in other words, you can say this is the male chromosome and this is the female chromosome. Another word we like to say, this is the mom, or this is the dad gene, a dad chromosome, and the mom chromosome, or mom genes and dad genes, whatever. All right. So in this way also, when they are in pairs, so for example, that the plant got one tall gene from this side and tall gene from the paternal and maternal chromosome. So both of them have the same gene. Yes, instead of writing tall and tall, we'll just use the symbols capital T and capital T, showing that they both are dominant traits, and we write them together. This, when we write two alleles together, is termed as the genotype. So this in words is said to be written as homo meaning same, and zygous. Zygous means pairing. Homozygous, and because they are showing the capital T, that means the dominant trait. So we write it down as homozygous dominant. So this is the first combination genotype. The second genotype could be on these chromosomes, you can either get tall and the other one could be short. So they don't have to be the same. Why? Because one is coming from, for example, in the plants, is going to be the pollen grain, and whereas the other chromosome, that is going to come from the female gamete, which is going to be the ova. So they're coming in from two different organisms, yeah. So they can be tall and short. How do I write them down? I'm going to use the capital letter T to represent tallness, yeah, and then the small lowercase t, small t, to represent the short allele. Both of these are termed as allele. In a genotype, we write down both the alleles together. So I hope you understand this by now. What's an allele? Allele is said to be all of the variants of a gene, all right, present on the homologous chromosomes. Now, so when you write capital T and small t, now how is this expressed in words? This is not homozygous because it's not the same. One is dominant, the other one is recessive. So this is termed as heterozygous, and that's it. The word hetero actually in Latin stands for the word other. Now the third type combination of the alleles on the homologous pairs could be that one gets the short allele and the other chromosome also gets the recessive or the short allele. How do I write it down? So I'm going to use a small t for the short one and again another small t to show the other allele. And this is now because they're both the same. So we'll use the same language, homozygous, and this is going to be homozygous recessive. So what we've just written down is actually the combination of alleles, the combination of alleles of a gene. A gene will be singular. Also, a gene can have different variety, different variants of alleles. So this combination of alleles of a gene, and this is termed as your genotype. I hope you understand the word now. All right. Now, if this is now going to be present on the DNA, on the chromosome as a gene. Now, this is not visible to us. What is visible to us as the physical form will be termed as the phenotype, right? So, if an organism has homozygous dominant combination having both the capital T, the dominant trait only, what will be, for example, here we're looking at the height. So what will be the, what do we say? What will be the height of this organism? Or if an organism has heterozygous combination of alleles, what will be the height? And the third variety again, if it's homozygous recessive, what will be the height? All right, so what do we know now? So you are what your genes are. You cannot go beyond that. You are what your genes are. If both of the alleles are capital T, or both of them are going to be dominant, homozygous dominant, then the plant would only be tall. There's no doubt about it. But if it is, and for example, if both of the alleles are recessive and both are short and short, so the organism cannot be tall. This plant cannot be tall. It will only be short. Alright, now comes this concept. And what do we say? We have both the tall and we have the short allele together. This is called the heterozygous combination. So what will happen? Will this come obviously in the physical type, physical form, or the tall one? Which of these? So here the concept of what we say complete dominance comes in, and the dominant allele will completely hide the recessive allele. Hence the what we say word, the title of complete dominance. So the height of the plant now will be tall only. Why? Because the recessive allele is recessive against this dominating one. This dominates the small t, right? So the capital T, because of that, the height of the plant will be tall. Okay? Now, but we'll add another word because both of the alleles are the same. So we say this is a pure breed or a pure animal for the genotype. Here, this is also a pure breed because both of the alleles are going to be the same. But this one is not pure because both of the alleles are there. They are different. So this is termed as a mixed breed. Another technical term for this would be that this is a hybrid. So this plant is a hybrid having a combination of both the alleles. Alright, now let's have a look at the genetic crosses of in monohybrid crosses. Okay, so what are the, first of all, let's have a look. What could be the possible combinations in our monohybrid crosses? All right, so let's see. So what do you mean by that? Well, we're going to take two plants, okay, and then we will cross them together, and then we will look at the probability of that trait being transferred on to their offsprings. Okay, so assuming this is, for example, plant A, and then this is another plant B, all right, and we cross them together. Now the word cross actually means that we will pollinate them or fertilize them together. So this is a term we use in genetics in which we mean that one plant is fertilizing the other plant, or another term for sexual reproduction you might say. Right, now plant A and plant B. Now what if this plant had this genotype and then this genotype, and then what will be the outcome? So we have to look at 4 crosses that everyone should know about, and the first one is that both the plants are going to be pure, but one is showing the dominant trait, right? So this is pure because it is having the same allele, and the other one has the recessive, the opposite one, and so this will be a tall plant and this will be a short. But mind you, this is a pure plant and this is a pure plant. So when they combine, we'll have to look at the outcome or the probability coming in the offsprings. What is, how many of there's a probability of them being, if one is tall and the other one is short, that means okay, what will be the probability of the offsprings being tall or being short. So that is where probabilities of genetic cross is coming. Now the second cross could be that I take one heterozygous and the other plant is also heterozygous. So what will be the ratio in the offsprings? And this is what we'll tell after doing the genetic crosses. Now number 3 would be that one is heterozygous and the other one is homozygous dominant, and again this is the pure variety. And the fourth one would be that one is heterozygous and whereas the other one is homozygous recessive. So we have to work out 4 basic genetic crosses to understand what this is all about. Now I'll take on the first cross and keep these crosses in mind. If you're writing them down, just copy them down and we'll work up the first one, then the second, third, and then the fourth. and then you can write down and compare the ratios in your copies. Now let's have a look at the first cross, right? So our cross number 1, cross number 1. The cross number 1 is going to be between two pure plants, and these are said to be the parent generation. Alright, and we wrote this down. The first cross was, both of them are pure, but one is dominant and the other one is recessive, right? So we write down the genetic cross for this. But there's going to be a certain format of writing down genetic crosses, and this is what most students have to understand, that you cannot change or alter the format. If you do it up in a messed up way, you will be losing marks. So you need to understand that everything has to be done in a certain way. Now, the first heading would be, on this side we're going to write down the headings, and on this side we'll work out the genetic crosses. So the first one is the parental phenotype. Now phenotype, like we discussed, phenotype is the physical form, right? So first of all is the physical form, or what we say the observable form. And the second heading will be for the same, what will be the genotype. So we'll just write them down here. The genotype, like we said, one is what? Pure, the other one is also pure, one is dominant, the other one is going to be recessive. So this is the combination. Now this plant, because it has tall and tall, so the height of the plant will be tall, and this one is short and short, so the height of the plant is going to be short. So we need to write this down as well because the headings are there. The third thing would be that these will undergo meiosis, and what will happen is that these two alleles will split, they'll separate out. So then comes the parental gametes. Now your understanding should be there that gametes are formed as a result of a special kind of cell division known as meiosis, and what will happen is that one of the chromosomes of a pair will now separate out. So what will you have? You will have each of these chromosomes or alleles, they'll come in one separate cell or one separate gamete. In gametes, we show them in circles. Like we said, there's a certain way of writing it down. All gametes are represented in a circle. Now these two can fertilize the gametes of the other plant, right? So this was our plant A and this is our plant B, and we're crossing them together. So this is one plant, pollen grain for example, fertilizing the ova of the other plant. Now these will only have this type of gamete, all right? Now in some of the books you can observe that you only work if they are the same, so you only take one gamete, all right? You just write down as a single circle, and this is the same, just say. In some of them you can take both of them. So let's see. Now after this, what will happen? Fertilization will take place. Now fertilization can be represented in a Punnett square. So the next heading would be that after fertilization will come in the second generation. So after the parent generation comes the second generation, and they are termed as offsprings. Offspring, another word for this is filial generation, all right? Filial. Filial means son, and offsprings is like children. So offsprings are genotype and phenotype. So we will be using a format termed as a Punnett square. Now Punnett square, Punnett was the name of a scientist, of a mathematician basically, and they devised a way of putting down information in a box. So let's write it down. So we'll make a box and we'll see what will be the outcome. So we make a box like this. All right, now how many gametes are there on one side? 2. So we'll write them down here, and we'll have 2 columns to work with. And how many on this side? There are 2. So we'll write them down as 2 rows. So this is here, and these gametes are written here. So I can write down the heading as this: gametes of one parent and this, so this is on one side, and this is going to be, if you want to write it down, gametes of the other parent of the one parent. Okay, so gametes of one parent, the gametes of the other parent. Now, how do you fill in the Punnett square? This is what is very easy to understand, but it becomes very, very clear. So write down the capital. So these headings, these letters, they will have to come in each of the boxes like this, and these have to come in each of the boxes like this. For example, capital T, I'll write it down here, and as well as here. And then this one also here and all the way here. Now you can remove the arrows afterwards, just for you to understand. Alright, capital T, and the same goes for these two, small t and small t. So each will go in each of the boxes, one and this and this. Alright, and that's it. Now in each box of the Punnett square, this is the genotype, and you will have to write down the phenotype or the physical form of that genotype in the same boxes. So this would be what? This capital T and small t, we have done this is heterozygous, and the capital T will completely dominate the small t, because again we're doing the example of complete dominance, right? So this organism, this probability will be of a tall height, again tall, because wherever you have the capital letter coming in, is going to dominate the other one. So this will be tall and tall. So after doing this, now what we have to conclude this thing, and how? So what do? We're going to write down the ratios. So this, after this is the genotype and the phenotype of all the offsprings. Now coming over here, so the first one would be, what are the genotype ratios of this Punnett square? Okay, and then if you look at the genotypes, this is heterozygous, 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 and heterozygous. So we'll just write it down over here. So we can say all are this, and in words it would mean all are heterozygous. And if you want to take out a percentage, and also we can just take out, say 100% are going to be heterozygous. So I'm just going to use the short form HET to represent heterozygous form. Done? So this is like a heterozygous. Now, okay, and you can also say out of probability out of 4 out of 4 all are going to be heterozygous. Okay, done. Now talk about the physical form of that ratio. So the physical form comes out to be a phenotype ratio. So what is the phenotype ratio? Now have a look, because all of them we decided had capital T, hence all of them would be showing the tall form, or tall, the plants would be very tall or having the tall phenotype. So tall, 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 and tall. So what do we say? We say all will be tall, and tall is also going to be of the, all are going to be tall, and they're all going to be not the pure, but they're going to be hybrid, if you want to say hybrid or the mixed breed, right? So this will be 100% tall, 100% tall, or showing the dominant trait. Alright, so that means the conclusion could be that when we cross, so the conclusion, if you want, you can write down a few words, and that actually becomes your question. Alright, so this is what the conclusion is, that if we cross, so this is like kind of a reverse question coming in at the end. If we cross two pure plants, two pure organisms, and one showing one of the dominant and the recessive traits, okay, then you might say F1, or you can say the word offspring. Then the offspring will show a genotype ratio of being all being what? Heterozygous. Alright, number 1, and the phenotype ratio being all being tall. Alright, so that concludes our cross number 1. Now going on to cross number 2, I leave the headings in their place, and then we can work out the cross. Now what do we do in cross number 2 is that we are actually going to take the offspring from the parent generation, and we're going to cross them together. What do you mean by that? The term we use in plants is self-pollination, all right, or selfing. We're crossing out the both of the same. All right, now cross number 2 will be that we will take two heterozygous organisms, right? We're going to take two heterozygous pea plants and we will cross them together, or you might say the offsprings of the parent generation. This is also termed as F1 self cross pollination. All right. So I hope this is clear. And what do you mean by that? Like the outcome we got in the first one was, so they had this, right? So what could be the headings stay the same, all right? So I'm not going to write them down again. You can just follow through. Now, the parental genotype of one plant, this is for example plant A crossed with plant B, they're both the same, all right? Again, so genotype would be what? They are going to be heterozygous into heterozygous, the first one. And now the phenotype, tall and tall, we just did this, tall and tall, 100%, they were all showing tall, even though they are the hybrid, even though they have the recessive allele. But this is a case of complete dominance, hence you can't see the shortness. Now the gametes, now this time this is going to be different. Why? Because when we separate them out after meiosis, and each of the allele will come into one gamete, so they'll be showing this. So they will be either be capital T or they will be small t, right? And same will happen in plant B, that they will either have this or they will have this. So write down a small or, right? Now after this, they'll be fertilizing, this will be fertilizing this. So for fertilization, we'll draw a Punnett square showing the offsprings, both genotype and the phenotype. So draw the Punnett square again. All right, and again we will write down the gametes of one parent on this side and the gametes of the other parent on this side. So this will be what? Capital T, and so these two come here and these two come on the side here. Capital T and small t, and this will be capital T and small t. Now how do I fill it up? I'm going to put capital T here and here, small t here and here. And this goes here and here and this goes here and here. So let's write it down. Capital T and capital T. Alright. Now this will be what? Small t and small t. Now remember, the capital letter, the capital allele will always come before the smaller case. So capital T, now I'm going to fill this up. So this becomes capital T and this comes here, this comes here. Same thing is here, small t and small t. So what do we now? This is the genotype, okay. Now what will the height, what will the phenotype be? So in each box, I'm going to write down the genotype and the phenotype. So this will be, because they have a capital T, this could nothing be but tall. This again, even though there's a combination, but this is a case of complete dominance. Just completely hide the recessive one. So this will again appear to be tall, even though it's a hybrid. This again, yes, it will again be tall. Wherever you have the capital T, that is showing the dominant trait, you're going to have the dominant trait. Now the last one, this one, small t and small t, what will this look like? Obviously this is short and short. So this will have a different variety in the phenotype, that will be short. Now let's write down the genotype ratios and the phenotype ratios for this. Right, let's go into detail. Now you got 4 combinations, so let's write it down. So we have capital T and small t, capital T and small t. So this is heterozygous. And then the last one is small t and small t. So this is what, this is homozygous dominant. These two are heterozygous. I'm going to use short forms. And this is again going to be homozygous recessive. All right. Now what is the number? This was 1 out of 4. This is 2 out of 4 and this is 1 out of 4. 4 meaning the probabilities that we had, all right? So we got this. Now in mathematical terms, this could also be written as in 1 over 4, 2 over 4, and that could simplify as half, 1 upon 2, and this is also 1 over 4. Now this would be in fractions, in decimals, this could be 0.25, and this is what, 0.50, 0 point this, and this could be 0.25, right? As a percentage also, you can go on and on, 25% or 50% and 25%. All right. Now, if we want to use a language, we'll say that if we cross a heterozygous with a heterozygous, then the genotype ratio in the offspring would be that they will be 25% homozygous dominant, 50% would be heterozygous, and 25% would be homozygous recessive. Or we can say, or the ratio is 1:1. Okay, now comes this is the genotype ratio, and mind you, all of these numbers are important because they'll be coming in your MCQs and your theory questions, and also your ATP paper, sometimes they come there as well. Alright, now the phenotype ratios would be what? Now have a look at the 4 boxes and write down the phenotype or the physical type again. 3 times, tall, 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 and then you have ratio short. So what is the ratio now? This is what? 3, ratio 1 only. And in other words, 3 out of 4 or 1 out of 4. 4 is the total number of squares, and we have this, the combination. So this would be 75%, and this would be 25%, or this could be 0.75, and this would be 0.25. So that is, now in language, what would we say? If we cross 2 heterozygous together, the phenotype ratio would be that there will be 3:1 to tall is to short, or 75% chance of a tall and only 25% of a short organism. Now 2 more crosses left, and let's see. Now cross number 3 and cross number 4. Now cross number 3 is heterozygous. Go back to the crosses. All right. So cross number 3. Cross number 3 would be a heterozygous and a homozygous dominant. All right. So let's write it down here. So plant A, for example, any of them could be either, would be this. Now both of them are tall, but mind you, one is pure and the other one is a hybrid. This is a pure tall and this is a hybrid or a mixed breed tall. Now the gametes would be what? Again, you split them up, put them in a circle. So the gametes on this side would be this and this. Now fertilization on the offspring genotype phenotype again shown in a Punnett square. Go like this. Write down the gametes of one here and here, and this comes out like this. I hope you're following through. Now see this. All right. Now each of them has a capital T, so all of them would be tall. All right. Now, so what is the genotype ratio? The genotype ratio is that it will be homozygous dominant ratio heterozygous, and the ratio comes out to be 1:1. All right. That means 50% chance of either. And you can also say that the parental ratio is maintained. The parental genotype is maintained. One is heterozygous and the other one is homozygous dominant. Now come down to the phenotype ratio. The phenotype ratio, what do we say? Well, all of them are going to be tall, right? So all are tall. So we say 100% will be tall because there's so many capital T's here, 1, 2, and 3. So there is no chance of the recessive trait coming forward. All right. Anyway, so this was our cross number 3. Now the last one. So each of these crosses, mind you, try to practice them again and again, because doing it once, this is just like maths. So the more you practice, even though you understood it once, the more you practice, the better it will be for you. Now cross number 4, and this is the last one for monohybrid. And what will happen, we'll take one heterozygous and the other one would be homozygous recessive. So if you write down the genotype, one is this and the other one is this, Now, what will they look like? What will be the physical form? What will be the observed form? That's called the phenotype. One would be tall and the other one is going to be short, right? Now, the gametes, again, you split them up. You separate out these two alleles like this and this again like this. So, one of them could have this, or this would be, all of them will be small t and small t. Now, this is going to fertilize this, and this we show in a Punnett square again. Okay, so the gametes of one parent written here, small t, and the gametes of the other parent written here, small t and small t. What is the outcome? Capital T, small t, and again we fill it up like this. Now see what you get. Okay, small t and small t. Now this is going to be tall, this is going to be tall because of the capital T, but this is going to be short and this is going to be short. So what, again, coming down, what is the genotype ratio and what is the phenotype ratio? The genotype ratio is that, yes, again, heterozygous, same ratio as the parents, heterozygous and homozygous recessive, and this is going to be 1:1 or 50% chance of either. And again, I'll just write this down here. Phenotype ratio is that out of 4, 2 will be tall, 2 probabilities of short, 2 is going to be for short. For tall, 2 for short. So we get both of the phenotypes in the offspring as against the first one. And this was what? 2:2 or 1:1. Again, 50% chance of either being, what is the word, tall. So there's an equal chance of the offspring getting up. So this is all about the 4 genetic crosses coming under the heading of complete dominance. The next lectures will be on incomplete dominance and codominance. Thank you so much.